Hey everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. I'm Professor Long, and today we're going to be going over some renal physiology. Quick disclaimer, because we are in the coronavirus shutdown, this course was originally a face-to-face course, traditional lecture, where I stood at a board, lectured, drew things out, and my students followed along in a note set that I published and helped them organize their information. We had to switch to online, and so I'm doing these videos rather quickly, rather crudely. They're all one-take videos, so if you hear any beeping in the background or any dogs barking or kids talking to me, I may not ditch a 25-minute video and start over, okay? So nonetheless, we've been doing renal physiology. In the first couple of lectures, we talked about the anatomy of the kidney, blood flow through the kidney, the pyramids and things. In the second lecture, we focused on the nephron, this structure here, which is the functional unit of the kidney, which is where we filter out all the waste, but we also pull out good stuff and other things, and then we have to put it back into the bloodstream. So you should know the functions of the urinary system. Essentially, it's to eliminate waste and toxins, but it can adjust blood pressure and blood volume. It does maintain the proper concentrations of valuable ions and nutrients. It conserves the nutrients, and adjusts blood pH. It helps regulate blood pH by getting rid of excess hydrogen ions. Urine is slightly acidic. Most of the body is pH 7.4. Urine is about somewhere around 6.5, so it's slightly acidic. Anyway, so when we look at the nephron, one of the things that we see is this would be our cortical radiate artery coming up into the renal cortex. We have the afferent arterial, and then as blood enters this little knot of capillaries called the glomerulus, that's where we do all the filtration. And we're going to begin today on page 90 with the filtration membrane. It's the second table on page 90 if you're following along in my note set. If you're not in my class, well, I can't help you with that. Now, the membrane there is pretty unique, and actually the whole nephron itself is unique for a couple of reasons, and I'll tell you why, but let's get through this. So we filter across that filtration membrane. We pull out water, we pull out valuable nutrients like glucose, amino acids, the breakdown of nucleotides. We also pull out some valuable ions, sodium, potassium chloride, calcium, a bunch of ions, as well as the trash and the toxins, all the ammonia-containing compounds, ammonia, ammonium ions, the amino group, which is a negatively charged ion, a lot of hydrogen ions, and sometimes some carbon dioxide and some urea. All of this trash that's being generated from metabolism is pulled out. So when we break down amino acids, we remove that amino group, something we call deamination in the first of the series of videos on the urinary system. And that deamination produces the amino group, which can combine with hydrogen ions to form ammonia and ammonium ions, all toxic to our body, can poison our cells. We don't want to consume that. It can shut down all sorts of things and kill you. So the primary function of kidneys is to pull all that trash out and get it out of the body. Now, one of the things I want to mention rather briefly is one of the big diet fads that's hit recently is this keto diet. And in the keto diet, you cut out all your carbs. And so that when we go into gluconeogenesis, when we run out of sugar to burn, our body starts to burn fat. And the goal is to burn a bunch of fat. One of the problems is you can start burning protein as well. And when you start burning and breaking down protein, you get a lot of deamination, and you can have all sorts of buildup of these ammonia compounds, and you can go into what's called ketoacidosis. So if anyone's trying those diets, I don't recommend it unless you consult a physician or a healthcare professional. But if you are doing those diets and you do them to a pretty extreme level, you better be monitoring your urine and your blood pH and a number of other things, because they are having some drastic effects on people's kidneys because of the overwork on the kidneys. Just a thought, because I know a lot of people are doing that stuff. So anyway, what we're going to do today, and by the way, as we filter out the filtrate and it enters the renal tubule, it becomes tubular fluid. As it goes through the proximal convoluted tubule, you should remember we pull out 100% of the nutrients back into the peritubular capillary bed, and therefore we retain them. We pull out about 70 to 75% of the water. Estimates change depending on whether it's cortical or juxtamedullary nephrons, but somewhere in there. And we pull out a variable amount of ions. When we get to the descending limb, remember the thick part of the tubule is simple cuboidal cells with microvilli. The thin part is simple squamous epithelium, the thinnest membrane in the human body, so we can move water and certain substances across easily. So as we get to the thin descending loop, we pull out a lot of water. That's going to concentrate the kidneys, or concentrate the tubular fluid, I should say. And it gets very, very concentrated in ions and waste. As we go back up the thick ascending limb, we have all these ion pumps that are going to pull the ions back out, which, by the way, makes this area very salty, causing the next volume of fluid coming through here to pull all the water out. It's osmosis and diffusion, basically. We get a lot of osmotic pressure because of all those salts. And then by the time we get to the distal convoluted tubule, we have a very concentrated urine, just a little bit of fluid with all the toxins. And we've maintained all those valuable ions and water and nutrients. By the time it gets to the collecting duct, it's going to end up at the tip of the renal pyramid, and in the minor calyx, major calyx, the renal pelvis, the ureter, the bladder, the urethra, and then out of the body. So we can pee out a small amount of fluid, but the huge amount of the toxins, all the toxins come out of the body. Now, what we're going to do today is we're actually going to walk through the renal corpuscle and what's happening there. And then we're going to go through the tubule and see what's happening in parts of the tubule. We're going to talk about tubular reabsorption and countercurrent multiplication again. And we're going to talk about the special things that happen at the distal convoluted tubule. So today's lecture is somewhat of a review of some of the details that we covered yesterday or in the last video. And we're going to go into a little bit more detail, okay? Now, some people want you to memorize all this osmolarity, 1200 milliosmoles, 900, 600, 300, or 300, 600, 900, 1200. I'm not going to get into the numbers on that. It illustrates a point, but I'm going to focus on what's happening on the physiology so that when y'all get to the healthcare phase of your careers and you're applying all this knowledge to real-life situations with pharmaceuticals and why are you giving this drug or why is this happening with the patient, you have a firm understanding. So now, we're going to start with the glomerulus, and we're going to talk about the blood vessels first. One of the things I want to point out before I even get to the filtration membrane is this, okay? Excuse me, I got to stand up. Everywhere in the body we have a common theme. We usually have a blood vessel that leads to a capillary bed, and as you know, capillaries are the only vessels that allow anything to enter or exit the bloodstream. They have the thinnest possible walls. That capillary bed is going to have pressure running across it. Remember, we did tissue perfusion and recall of fluids in the dynamic center when we did circulation. Well, that's what's happening here. As the pressure is really high, because this is our arteriole coming in, then we have a capillary bed. They call it a capillary bed because it is a whole bunch of little vessels that break up, almost like a flower bed. If you made a bunch of trenches and ran water, it would flow through all the trenches. Kind of like when you see a field of crops when you're driving out of town. So, and then along that capillary bed, we have a dynamic center where we start to get recall of fluids. And then we have a venule. It's a common theme in the human body. And we have these all over the body where we're flowing blood that's rich in oxygen and nutrients and ions in here. We get tissue perfusion, all the good stuff comes out and bathes the cells, and they bathe in their glorious goodness and get all their nutrients and nutrition, but then they dump their waste in it, but that waste goes right back into the same capillary where we get recall of fluids. And then it hits the venule. Well, the kidneys have one unique feature, and one of the things we know when we look at anatomy and physiology is whenever we have a difference that's unlike everywhere else, you got to ask yourself why. What is the point? Well, when scientists saw in the kidneys, we see this. Now, I'm not going to make a neat, pretty capillary bed. I'm just going to do some skinny little lines. But we have a capillary bed here where all these crisscrossing vessels come together. And then we have another capillary bed. Then we have the venule. And if you notice, there's no dynamic center in this first capillary. This is going to be our afferent arteriole. I'm using the abbreviation I used last time, so if you didn't watch the last video, it may not make sense, but AA stands for the afferent arteriole because it's coming into the glomerulus. This is our glomerulus, this is the efferent arteriole, and then this is the peritubular capillary bed, okay? Why do we have this difference here? Well, One of the differences about the glomerulus is you notice there's no dynamic center. The blood pressure in the glomerulus is higher than the average capillary. And the reason that's important is because as I raise the pressure down here, as we raise the blood pressure on one side, remember that pushes the dynamic center further towards the venule. If I raise it high enough, it completely eliminates the dynamic center, and I would not get any recall of fluids. And that's exactly what we want right here, because I need to filter out all of this filtrate that has all my good stuff and all my bad stuff, and I don't want any reabsorption here, because I could possibly reabsorb some of the trash and toxins and they would stay in my bloodstream. So I need the glomerular pressure to be higher than the average part of the human body, than the average capillary bed, to eliminate the dynamic center, so I simply get tissue perfusion. And really, we don't even call it that here, we call it filtration, because we're filtering across this membrane. Now, as substances flow through the tubule, we're going to pull all the good stuff back in to the peritubular capillary bed. We want the dynamic center there. So this is basically a normal capillary bed that has a normal dynamic center. Actually, the dynamic center on this one is a little bit further over, but nonetheless. One of the other important distinctions is this. When we studied blood vessels anatomically, what is the difference between an artery and a vein, or an arteriole and a venule? Well, when we looked at their walls, if you recall, arteries have a lot more smooth muscle in them, so do arterioles. And then they have the tunica externa and interna, but we're not going to focus on that. If I were to look at a vein, the venule doesn't have a whole lot of musculature, hardly any at all. And what that allows is arterioles to constrict and dilate. If I constrict this vessel, the smooth muscle contracts, the lumen gets smaller, less fluid can flow in, so therefore I've decreased the amount of fluid flowing into the glomerulus, or I could be stopping up the efferent arteriole. So imagine if this is my afferent arteriole and this is my efferent arteriole. If the afferent arteriole is dilated and the efferent arteriole is constricted, then I would have a lot of fluid starting to build up and create a lot of pressure in the glomerulus. So I can raise the glomerular pressure by dilating the afferent arteriole and constricting the efferent arteriole, holding fluid in there, getting proper filtration. And our body has the ability to do this. We're going to talk about this at the very end when we talk about regulating our filtration rate. But our body has the ability to do this if, let's say, your blood pressure is dropped pretty low for a long period of time, then we can adjust. We have to remove toxins at a constant rate, because our body is always metabolizing. Whether we're feeding it or not, it's going to start digesting itself. And that's going to produce toxins and waste. If we do not rid the body of the toxins and waste, over time the levels start to build up, your blood goes septic, and we begin poisoning our cells. And the biggest wimps are the brain and the heart. They start dropping first, and then you shut down. Now, if blood pressure is low, I want to dilate afferent, constrict the efferent. So it's important to have two arterioles here. Now, what if my blood pressure is high and I'm filtering so fast the fluid is just flying through my glomerulus, and those little cells in the proximal convoluted tubule and along the loop of Henle aren't able, not the cells, but the proteins aren't able to transport all the good stuff back in? Then some of it could flow out and I could lose valuable nutrients and ions. So in order to avoid that, when our blood pressure is elevated, like under sympathetic stimulation or long periods of stress, now I can take the afferent arteriole and I can constrict it, and I can dilate the efferent arteriole, and that will relieve the pressure that's building up in the glomerulus. So I can raise and lower the glomerular pressure, or I can adjust it to remain the same, even if blood pressure goes up or blood pressure goes down. If blood pressure is low and blood volume is low, then I need to dilate the afferent arteriole, constrict the efferent. If blood pressure is high and blood volume is high, then I can do the exact opposite. Sorry, I put an EA there. I can, oh no, wait, sorry. I did this wrong. Let me do it this way. Okay. I'm confusing y'all and I'm confusing myself. Afferent arterial, efferent arterial. So this would be if blood pressure is low and blood volume is low, then the afferent arterial is dilated, the efferent arterial is constricted. If blood pressure is high and blood volume is high, then I constrict the afferent arterial and dilate the efferent arterial, and I can keep my glomerular filtration pressure constant. Okay, we're going to come back to this concept in a little bit, but that's an important concept as to why we have that. Now, what we need to look at is a few things. Okay, so let me erase all of this, and we're going to do this again. I know I kind of botched that, but that's all right. I wasn't planning on doing it at this time, but it just popped in my head. So you need to know that that's why we have two capillary beds here. This one lacks a dynamic center for filtration. This one has it for reabsorption. Now, back to page 90, the filtration membrane. So if I could take a little piece of this capillary and cut it out, and just draw a little piece of it here, like this, what I would see is it is a series of simple squamous epithelial cells, very thin here. This would be the cell membranes of all these epithelial cells, kind of all sewn together and stuck together. Now, in between some of these cells are these openings, or fenestrations. And sometimes between the membranes, we have these little openings. Here's the concept that I like to get across to students to try to explain this. Imagine if you took a long cardboard tube, like the ones that Christmas wrapping comes in, or we took a long plastic tube, but similar to that, and we poked a bunch of holes in the plastic. So I have this long plastic tube that has holes all the way down its length. And the holes are pretty good size. They're everywhere. If I try to run fluid through this tube, then you would notice that that fluid starts to drip out. Okay? Now, what if I take this fluid and I dissolve some sugar? Take a big couple of scoops of sugar. I take some amino acids like some protein powder. I put in there some salt. I put in there a bunch of waste like ammonia and ammonium ions and things. So now I have all these molecules. And I throw in some protein. Maybe I grind up some steak or something. And I put all the structures in here. Some BBs and some marbles. BBs would represent red blood cells. Marbles would represent white blood cells. So now I have red blood cells in here. I have white blood cells in here. Well, they're far too large to fit through the holes. And if I do this with the right pressure, some of the water will come out the other end, and I'll have my cells in there. Now, I have also mixed in here large proteins. Large proteins like antibodies and other things. Well, it turns out the large proteins are actually small enough to fit through those holes. And I could be losing a lot of large proteins. Maybe I have some smaller proteins in here too. Well, if the large proteins can fit through the fenestrations, so can the small proteins. Like albumin is a small protein. Now, albumin is extremely important for osmotic pressure of our blood. We'll talk about that in some more detail in just a second. And then I have all my nutrients. So let's say these little green dots represent nutrients, which are way smaller than the proteins. So I've got glucose and amino acids and ions and other things in here. And then finally, I'm going to have my toxins in my waste. Ammonium ions, ammonia. These are also small molecules, and they're going to be in here. I don't even know if you can see these dots, but you get the idea. Now, this is going to be the filtrate that we're forming across that membrane. So one of the things we know is the first layer of the filtration membrane is the capillary endothelium, the blood cells, the squamous cells, I should say, not blood cells, the squamous cells that make up the lining with the fenestrations. It is permeable to large proteins, small proteins, nutrients, ions, waste, water, all that would come dripping out here. Now, I cannot afford to lose the large proteins. Proteins are valuable. Getting protein, for us, it's pretty easy for most of us, but if you were to live in the bushland in South America, or if you live in the mountains in Peru or in Africa, or the Aborigines in New Zealand, Australia, or wherever, some of them live in a very rural agrarian society. They don't know where the next meal's coming, so they don't get enough protein sometimes. Or if you watch Naked and Afraid, they're always dying for protein. So we cannot afford to lose protein. So now, what if I took my tube instead of a blood vessel? But what if I took this tube and I took, the little metal stuff that makes up the filters, or if I took like an air conditioning filter, the plastic inside there, the crisscrossed film, the mesh is what I'm looking for. That's the word I'm looking for. If I took that mesh and I slid a sleeve of it, or like a sock or fishnet stockings, if I slid that over here, then the crisscross fibers here would actually block up part of these fenestrations. And as they crisscross here, it makes much finer, smaller holes. Well, this is going to be what we call the basal lamina, which is basically like the basement membrane, okay? And it's also sometimes referred to as the lamina densa, because this one's a little bit thicker, but it's a basement membrane. Always where we have epithelium, we usually have a basement membrane. There's a rare exception. So now, these holes are so fine that not even the large proteins fit through there. But the small proteins like albumin and other things can. So I don't even want to lose the albumin. So Mother Nature, in her grand wisdom, had us evolve these cells called podocytes. Pod means foot, from pata if you speak Spanish. And these podocytes have these little foot-like structures that extend over the cell, or over the capillary. And they'll have their little nucleus here, making them a little bit bigger. But if I color in my podocyte here, you'll notice that his foot pods actually cover these fenestrations, and they'll cover a lot of the holes here. Now, if I took another podocyte, and I'm going to make a different colored one, okay? If I took another podocyte on the other side here, and I wrapped him around like this, and his little foot pods came down in between these, you can see how they very tightly cover all of these fenestrations. It's almost as if I take two hands, and I'll do this. Well, I don't like to do it because I'm going to be writing and you're going to be staring at it, but I'll do it anyway. If I put a nucleus of a cell here, and I put a nucleus of another cell here, I have two podocytes. These little extensions are called pedicels. If I interlock the pedicels, then the gaps between them become very tight. Now, if I poured water in my hands, the water would come dripping through, but anything else in there that's pretty large would not. Now, little tiny things like ions and glucose and amino acids would come dripping through. So we call this, if this is the capillary endothelium, then you can see where my pedicels are of a podocyte. You can see the blood vessel here. And then if I wrap two of them together, you can see that I can make very tight filtration slits. So that's the third layer of the filtration membrane. The filtration membrane has three layers. Capillary endothelium with fenestrations that allow large proteins, small proteins, and then all the other stuff in the filtrate through. We don't want to lose large proteins, so the basement membrane, the lamina densa, has proteins in it, protein filaments, and the crisscross fibers in it that prevent the large proteins from permeating. So it's not permeable to large proteins, but it's permeable to small proteins and everything else. And then we have the podocytes. The podocytes interlock their pedicels, making these very fine filtration slits that prevent even the small proteins from dripping out. So now, when I form a filtrate out of that filtration slit, it has my toxins in it, it has nutrients and ions in it, but no more protein. And that's what's in this table on page 90. It tells you the capillary endothelium is permeable to large and small proteins, the nutrients, ions, and water, but it's not permeable to blood cells. The lamina densa is permeable to small proteins like albumin and other things, but it's impermeable to large proteins and everything larger. And then the filtration slits, or the slit pores formed by podocytes, are permeable to the nutrients, the ions, the water, and the waste, but they're not permeable to proteins. This is why we don't lose a lot of protein in our urine. If you do have protein in your urine, by the way, it's a concept, or it's called proteinuria. It means protein in your urine. Not a good sign. It means there's probably something wrong with your filtration membrane. If you have blood in your urine, they call it hematuria. Oops. You don't want blood in your urine. There's something wrong with the filtration membrane. We have damaged the glomeruli, and we're losing valuable substances that we can't afford to lose. Now, I hope that makes sense to you and you understand that concept. It's kind of a long way to get there, but I wanted to explain all of this, all right? Now, I'm going to erase all of this, and we're going to do the next concept, and then I'm probably going to stop this video, and we're going to move to the last part, all right? These videos do get kind of long because I take my time to explain things. Anyway, we're going to talk about filtration pressures, okay? And the reason we're going to talk about the filtration pressures, and this is going to bounce between page 91 and then back to page 90. I'm going to do it a little bit out of order. But filtration pressures are important, and it's going to show you the importance of some of the proteins in our blood. So there's an equation that we can talk about called the net filtration pressure. Okay, I'm going to switch colors because this marker makes a lot of noise. The net filtration pressure is based on two other pressures. It's based on one called the net hydrostatic pressure minus the blood colloid osmotic pressure. Okay, so net filtration pressure, net hydrostatic pressure, blood colloid osmotic pressure. So let me explain this one first. This is going to take us to page 90. The net hydrostatic pressure. If you remember, whenever we have a tube, if I'm pumping fluid through the tube, the fluid is pushing out against the walls. That fluid pressure, that pushing pressure, is what we call hydrostatic pressure. That's a pushing pressure pushing out on the walls. That would be trying to form filtrate. If I had holes in the walls, that would be squirting stuff out, kind of like a sprinkler, okay? And when you turn the water up on the sprinkler and increase the pressure, it squirts more fluid out. If you turn it down, it squirts less fluid out. So that's the hydrostatic pressure, that pushing pressure. As you should know, dissolved in your blood, we have a whole bunch of protein. And the most primary protein that's involved in this is a protein called albumin. Albumin is what makes the white part of the egg kind of slimy and viscous. And if you go back to part 1 A and P, let me draw this out real quick because this is a concept you can never lose sight of. If I have two chambers separated by a semi-permeable membrane that has holes in it, I put some substance on one side, dissolve it into the fluid, and I put a little on the other. Let's say this is albumin. Let's say this is plasma, and this is the filtrate. Okay? If these proteins could cross that membrane, they would, by the laws of diffusion. If they cannot, then they pull water back across this way. And so we have this blood colloid osmotic pressure. It comes from the word colloid, which means a suspension, a thick viscous fluid. This osmotic pressure is created in our plasma by albumin, and it's trying to pull stuff back into the bloodstream. So I have these two fluid pressure, or these two pressures fighting each other. Now, we're going to come back to this in just a moment. So let me get to the net hydrostatic pressure. So the net hydrostatic pressure is going to be this. If you've ever seen, well, let's say you were filling a jug with a faucet, okay? So I have like a milk jug here or a large vessel, and I have a hose going into here, and I have fluid draining. As it begins to fill up the vessel, as soon as it gets all the way up here, it starts pressing, and this fluid stops draining if these two pressures become equal. Well, this pressure pushing down is going to be called the glomerular hydrostatic pressure. That's the hydrostatic pressure inside the glomerulus. This is going to be the capsular space, the space inside the Bowman's capsule, and then I would have the proximal convoluted tubule that can drain it. And so this is going to be my capillary, I'm sorry, I did that wrong. Yeah, it's the glomerular hydrostatic pressure, and this is the capsular hydrostatic pressure, not capillary. because that's the pressure inside the Bowman's capsule, outside the blood vessel. If these two pressures are equal, I'm not going to filter anything. I need my glomerular hydrostatic pressure to be greater than the pressure in the capsule so that I'm forcing fluid into here. So that's how we develop what's called the net hydrostatic pressure. The net hydrostatic pressure is based on glomerular hydrostatic pressure, which is essentially our blood pressure, and the capsular hydrostatic pressure. When you do the math, you get net hydrostatic pressure. Okay? Now, that's a lot of talk about something that I'm probably not going to test you on. Okay? I'm not. But I do want you to understand this part. So, my net hydrostatic pressure simply means that the pressure in the glomerulus is higher than it is in the capsular space, and I am filtering. Okay? Now, I also have the blood colloid osmotic pressure trying to pull back into the capillary. So all three of these pressures are kind of fighting each other, and what we need to know is this. If the net hydrostatic pressure is greater than the colloid osmotic pressure, in other words, if net filtration pressure is positive, we are filtering blood and cleaning it. If it is 0, then we are not filtering. If it becomes a negative number, we can actually reabsorb. We could develop a dynamic center in that glomerulus and start to reabsorb through recall of fluids some of the toxins. So if blood pressure remains low for a long period of time, then I would actually not be filtering all the toxins out of my blood, and my blood could go septic. If blood pressure is high, I would be filtering. The problem is if I filter too fast, then the fluid would be being squirted through here so rapidly, I may not be able to capture all the ions before they escape. Okay, so we're going to come back to this concept, but I wanted you to understand that on page 90, and really I should have put it on page 91 as well, if either of the pressures, net hydrostatic pressure or net filtration pressure, if they're positive, I'm filtering. If they're 0, nothing is entering or exiting the blood vessel. If they're negative, I'm going to be reabsorbing trash back into my bloodstream. So we need to have a positive net filtration pressure. If it stops filtering, we're in renal failure. The kidneys have failed to do their job. And all these pressures are fighting each other. Which, by the way, is why we must maintain adequate levels of albumin and protein in our blood. You have to have protein in your diet in order to replace these albumins to maintain a good blood colloid osmotic pressure, which affects blood pressure and blood volume. All right. There are some questions on page 91. You can look up those questions. You can look up the glomerular filtration rate. And I'm going to stop this video right here because it's starting to get a little bit long. Actually, you know what? I think I'm going to knock it out because it's going to take me about 10 to 15 minutes. So let's do this. Let's just finish instead of you having to watch a whole new video. All right? So a couple of things you should know from this video. Why do we have 2 capillary beds? So that we can have filtration occurring at one, reabsorption occurring at the other. You need to understand the structures that make up the wall of the filtration membrane, the 3 layers, what they are permeable to. You need to understand that albumin plays a huge role in blood colloid osmotic pressure and blood volume, and we cannot lose proteins, we cannot lose blood in our urine. So now we're going to go into the tubule. We've talked about this. Let's go into the tubule itself. Tubular reabsorption. Okay. Actually, before I do that, let's do regulation of, let's finish up with the glomerulus itself. Okay. So I talked about the afferent and efferent arteriole. So the afferent arteriole comes in. We have the efferent arteriole coming out the other end of the glomerulus. And we talked about the size. This is a concept referred to as autoregulation. The kidney itself has the ability to monitor some pressures. And from one of the models in lab, you'll see this. Where the afferent arteriole comes in and forms the glomerulus, and then the efferent arteriole comes out, afferent and the efferent arteriole comes out and forms the peritubular capillaries. There's a section that's a cutout on the model where we see these little kind of yellow looking cells or tan looking cells that make a little ring over here. And one of the things none of our diagrams really shows us is this. If I were to have a glomerulus sitting here like this, with the efferent arteriole coming out, it turns out it's not all laid out like this, all nice and pretty. What happens is the Bowman's capsule sits here, and then we have the proximal convoluted tubule all knotted up. That gives it a lot of surface area for reabsorbing things. And then the loop of Henle comes down, and I'm going to change colors just so that you can follow this. Then we have the distal convoluted tubule come up, and it's knotted up right here next to it. Sometimes they look indistinguishable. But one of the things that happens is a piece of the distal convoluted tubule will loop over here before it goes to the collecting duct. And so if I cut this little section out, I'm going to get the afferent arteriole, the efferent arteriole, and I'm going to get a piece of the distal convoluted tubule here. And on one side, the cells are a little bit thicker than they are on the other side. So on the model, we call this little group of tan cells right here, if I can get that open, that's called the juxtaglomerular cells. And this thick part of the distal convoluted tubule is called the macula densa, means the thick spot. Those two cells are monitoring the pressure of fluid coming in and the pressure of fluid over here. And if those pressures are not right, then there's a mechanism where they can affect the afferent and efferent arterioles. If they detect that filtration and blood pressure are low, I'm going to dilate the afferent arteriole and constrict the efferent arteriole to restore that pressure so that my glomerular filtration rate remains constant. So I don't want my glomerular filtration rate to drop if blood pressure drops. So the kidney has the ability to make its own adjustment by dilating and constricting the afferent and efferent arterioles. The reason it's important to have two arterioles, because if there was a venule here, we could not do that. Now, if blood pressure is too high, then I would be squirting stuff out of here so rapidly that it would fly through here and I would not be able to recall all the nutrients and ions. So I can constrict the afferent arteriole and dilate the efferent arteriole. Now, I did this for you a few minutes ago, and that's why we said if blood pressure is low and blood volume is low, then our glomerular filtration rate could drop, so the afferent arteriole will dilate. No, it won't. It will constrict. No, if it's low, afferent arteriole dilates, the efferent arteriole constricts. If blood pressure is high and blood volume is high, then the afferent arteriole will constrict and the efferent arteriole will dilate. Okay? So you need to know this little table. Blood pressure and blood volume are low, the afferent arteriole can dilate, the efferent arteriole will constrict, and that will cause the filtration rate to go back to normal. And they will adjust until it's just right. If blood pressure becomes too high, then I have to constrict the afferent arteriole and dilate the efferent arteriole. There's a lot of terminology there. So anyway, you need to understand that, and that's called autoregulation. Okay, there's three ways we're going to regulate the glomerular filtration rate. And my note said on the top of page 92, next to autoregulation, the number 1 did not print when I sent it to the printer. Sorry. The second way is hormonal regulation. These cells also, since there's some pressure or baroreceptors there, can also act as an endocrine organ, or at least they release an enzyme that sets off an endocrine pathway. So one of the things that can happen is if we don't have enough pressure here, there's some feedback in the brain that will tell us to release ADH. ADH will target the distal convoluted tubule and increase water reabsorption. As I pull more water into the distal convoluted tubule, I'm going to raise my blood volume and my blood pressure. Also, the juxtaglomerular apparatus, which is just these cells and these cells combined, can release the enzyme renin. And as you know, our liver is constantly releasing angiotensinogen. Well, as soon as I release renin, it converts angiotensinogen to angiotensin 1. Angiotensin 1 goes through the lungs. Angiotensin converting enzyme, ACE, converts it to angiotensin 2. That binds to cells in the zona glomerulosa of the adrenal gland, and that releases aldosterone. Salty aldo targets the distal convoluted tubule and increases salt reabsorption. By osmosis, water follows the salt, raising blood pressure and blood volume. So that's the hormonal regulation. Actually, the third hormone would be atrionatriuretic peptide, which does the exact opposite of aldosterone. If our pressure in our right atrium is too high, we have too much blood pressure, then the right atrium releases atrionatriuretic peptide. It targets the distal convoluted tubule and causes us to lose salt and water, lowering blood volume and blood pressure. So that's two of the three ways. One is autoregulation, afferent and efferent arterioles adjusting pressure, the glomerular filtration rate. The second way is hormonal, three hormones that we're going to talk about for this test: ADH, aldosterone, and atrionatriuretic peptide. Know all three. And then there's autonomic regulation. The autonomic nervous system is sympathetic, parasympathetic. When we are stressed, we tend not to urinate as frequently. It messes up all our waste functions, defecation and urination. You don't defecate as often, you don't urinate. That's why your doctor sometimes asks you, have you had any changes in your going to the restroom, your restroom routine, how often you pee, how often you defecate? So when we're stressed, the hormone adrenaline, that we also abbreviate as norepinephrine, and even epinephrine, if you take a whole bunch of it, can target the afferent arteriole and constrict it to a bare minimum, which would decrease your glomerular filtration rate, which would decrease urine formation, which stops you from going to the bathroom as often. Okay, so that's the autonomic regulation. Sympathetic neurons can release adrenaline and constrict the afferent arteriole, shutting off or decreasing urine output to a bare minimum, which is why prolonged periods of stress are also not good, because you're not ridding the body of toxins. Anyway, so those are three ways that we regulate filtration at the glomerulus. Okay, now we're hitting about 40 minutes on this video, so I'm going to take a stop there. In the last video, we're going to go through the tubule, and we're going to talk about some of these details again. I hope you learned something. I hope you had as much fun as I did. And I hope you guys keep trucking and don't lose your faith. I know this semester is winding down. I know we have a short window of time to get all this done. Please keep pushing, keep plugging. There's nothing we can do about the circumstances that we're under. We cannot change the cards we're dealt. We can only change how we play the hand. And you can sometimes, if you watch gambling on TV, you see people win with 2 deuces. So, you know, it's not the cards you're dealt that determine whether you win or not. It's how you play your hand. Or as I always like to say, you cannot change the wind, but you can adjust your sails. So let's have a positive attitude. Let's try to finish this semester and get through all of this. Again, sorry this goes so long, but I'm trying to get the information across. I could be much more quick, but I really like to take my time to explain it so that you can follow it and simplify these difficult concepts and then understand the more difficult stuff from a simplistic viewpoint. All right? Thanks for watching. See you guys on the flip side.